So our last session of this workshop is a panel discussion, and we actually are very happy to have our panelists up here with us to discuss more about mRNA process development and manufacturing. This panel discussion will focus more on the future outlooks and the challenges that we've faced through tech transfers and some of the strategies that our speakers has previously eluded. So to start, so I'll just do a quick intro about me and the panelists. So my name is Jimmy Chu. I am an associate director at 2seventy Bio, working on mRNA process development, and I'm actually the moderator for this panel. So I'll be mainly asking questions and picking audience for ask questions and moderate the sessions. And we have three panelists first joining us today. Two of them you already see this afternoon, Aaron and Mukunda. But we have a third panelist joining us, Molly, and I'll let Molly to give an introduction about herself and her company, and then we can start the panel.
Thank you, Jimmy. So I'm Molly Mcglaughlin. I'm the Chief Operating Officer of Kudo Biotechnology. Kudo is a relatively new company. We are a contract development and manufacturing organization headquartered actually in Needham, Massachusetts, with a global footprint in Shanghai and also in Singapore. But we are an end-to-end mRNA CDMO. So we do everything from cell line development for your plasmid through production and purification of plasmids, gene of interest, or we can actually do AAV plasmids as well. And then we do the IVT transcription, and then also LNP encapsulation and formulation and sterile finish fill. What's different about Kudo is we do this all under one roof. So instead of managing 5, 6, 7 different contract manufacturing organizations or CROs, you can come to us and we can do the full service of everything from sequence all the way through clinical trial vials, packaging, and shipment to your trial sites with all the regulatory analytics for full release as well. So it's a new organization. We are carved out from a product company that decided to get out of CMC. So the team and the manufacturing is actually extremely experienced. We've done multiple GMP batches that are in up to phase 2 clinical trials globally. So like I said, it's a new organization, but we're very happy to be speaking on this panel today and discussing the future of our modality.
Great, thanks, Molly. And we will let the two other panelists just quick intro who you are, your title and function and company before we start.
Yeah, so as Jimmy introduced earlier, I was a global head of global director quality of external engagement. That's actually title has changed yet again. And since I put president, I'm actually Australia assurance and compliance expert in the role. So as I mentioned to others, the pandemic did a number on a lot of people working at Moderna, and a few of us kind of looked and said, I don't really want to do that again. So moved into something that wasn't so fast paced, but still using my skills and ability. But my background is a chemical engineer, process engineering, MS&T roles for a few years now.
My name is Mukunda, Krishna, Associate Director at Arcturus Therapeutics, responsible for manufacturing at our external CDMOs and also the supply chain, procuring raw materials across the board from be it your cell banks or DNA or mRNA or LNP.
Great. So to start, I think maybe I'll ask the panelists about what have we come so far in terms of mRNA therapeutics and the mRNA manufacturing side. What have we accomplished? I think given the examples of the 2 mRNA vaccines, we've come through a lot. So maybe have the panelists give their thoughts on what we've accomplished so far that's been beneficial to the whole field.
I can go. I mean, I think obviously, when I looked at, so I've been with Moderna for a little over 6 years now. when I was looking to move into Boston, I was actually leaving a commercial manufacturing facility running flu, and like I mentioned, 5,000 liter bioreactors making 50 grams of antibodies. Just if you think about it, that's a ridiculously small amount at the back end. So when I started research to where I wanted to go work for, Moderna was there, and I did a little research. So I went but went from commercial, support type function back into a clinical, support function, but really saw mRNA and as much as a chemical engineer can understand the biologics of it. I was looking at that going, wow, you mean the body's going to make the proteins? I'm like, that seems really, that's really impressive if it works, right? And so, I think that for me, if you look at that, is like at that point in time, Moderna had just started a phase 1 clinical trial when I started there, about a year into a phase 1 clinical trial. Had a number of candidates in the pipeline, and what we saw, I think, from the safety aspect of the mRNA is really where I think that was the important driver. That's important outcome of the pandemic, was now you went from having, let's say, hundreds, maybe even thousands of patients to millions, right? Billion, right? Worldwide doses. So that accelerated that safety of mRNA and the understanding of mRNA. And obviously the lipid family is a big part of that. So there's still a lot of work for other companies to do, but that's driving that acceptance of it, if you will. And I think that's public, getting public opinion on the right side of things is as important. As we've known in vaccines for years. We've had to fight that for years. So I think getting the public on the right side, and hopefully getting it out of the political space would be nice, but that'll happen sometime down the road. And then I think it will really see a progression of if you take monoclonal antibody progressions, you take industry progressions, I expect to see a significant shift, if you will, in my opinion, in the acceleration of new drugs or new vaccines and new therapeutics to market.
Yeah, over the last 3-4 years, I think like a lot of us have learned quite a bit about how you can make this molecule, right? It's key to understand, like, the scientists are able to make this prototype or an example of what needs to be done, and over the year and a half or so, we're able to like produce that at scale. You can go ahead and utilize it for a good cause, and to be able to like prevent disaster or reduce disaster across the world. And that, I think, is the progress of and the power of science, to be able to like achieve that in such a short amount of time. I think that's where people coming across from different places to make that happen, that's the power of mRNA.
And I would agree with Aaron. I think that mRNA has become a modality that's proven. I think the COVID vaccine has proven its not only its safety profile, but its regulatory path. So, I mean, I've been in oligonucleotides and antisense for quite some time in my career on the CDMO side, as well as viral vector. And I always thought mRNA where today was like where our viral vectors were 10 years ago, but I think it has accelerated it. But I think the regulatory path, knowing what it is now and how it can be accelerated, has really proven that this is a real modality, not only for infectious disease, but also for therapeutic indications as well. And I think that we're going to see the future of mRNA be very promising as a result of that.
Great, thank you. And I think the regulatory path is actually a very good point, and maybe touch a little bit on that. I think initially, the regulatory, there's a little knowledge on how we can get through the path, right? Now with more understanding, do you anticipate, and this is for all panelists, do you anticipate an easier conversations or regulatory package? Or do you anticipate, even more sophisticated questions or questions that are more, critical to the development for your product?
No, and I can touch on that a little bit. So during the pandemic, I'd say October, September, something like that, 2020, the FDA came on site for a, what's the right word, visit, unofficial visit, basically, not a 482, just an unofficial visit. They started the first day was like a traditional inspection, which we needed, and it was good to have that. But then the next day was like, okay, the hats are off, let's talk. And so it was an opportunity for us to get because I don't know how many of you have been through health authority inspections. You usually don't get your vice president of R&D in front of an auditor. That just doesn't happen. You don't do that. So we did that, and so we that opened up a conversation, I think, for us with the FDA, and that I've never experienced before. I've never experienced the kind of transparency that Moderna and the FDA, and it's both ways. Moderna and the FDA have worked together. I think that was the difference, right? They wanted to understand our process. We wanted them to understand our process. So I think that's something you're seeing a much different. I mean, and I don't know the other industries, cell and gene therapy in general, but at least for mRNA, they wanted to work with us. And they wanted to understand it. And even the first inspection we had, the FDA doesn't normally send a reviewer. So the reviewer is the person that sits and reviews the application. They usually don't send reviewers with the inspection team, but they did because they wanted to make sure that guy maintained his knowledge, because he was a reviewer and that person was a reviewer. So, but I think where the biggest challenges we've seen is sort of, especially as we talk about ATMPs, mRNA from a vaccine is not an ATMP, but mRNA for a therapeutic is an ATMP, and that creates a little bit of a headache for us. So I think there's still a niche that needs to be created and a pathway, so to speak. The other example I'd use is BioNTech, Pfizer. They filed their LNP drug substance, what we call our LNP drug substance. They filed it as a drug product intermediate. We were able to fight it down for a little while, but now recently we had to file it as a drug product intermediate for the EMA, but not in the U.S. So now we've created another disconnect that we'll have to figure out how to undo that in the future. But our experience has been really, I would say, from all the health, I was involved with most of the health inspections for the Norwood site. So TGA was there, EMA, Canada, Health Canada, UK. So I got an opportunity to kind of be involved with a number of groups, and they were all much more transparent than what I had been used to in wasn't hostile environment that I'm used to with inspections. I don't want to say be negative, but my previous experiences was somewhat hostile environments for inspections.
Yeah, I think the regulatory pathway is going to be interesting as we move forward to scale up and manufacturability and proving different than the platforms that we have for the Moderna and the BioNTech Pfizer product. I think that as you get it to different, whether it's a PCR template versus a plasmid template, whether it's a mixture of two RNAs into one LNP, whether they're going to question how can you really quantify your empties and full LNPs. I think that it's going to, as we get into other than those two platforms, I think the FDA is going to get more and more critical about the manufacturability of these products. And so we should anticipate that and make sure that we're prepared to answer the hard questions on how do you quantitate that this is what you say it is.
Yeah, for us, it's most of the time trying to explain that we are a little different from the Moderna and the Pfizer and BioNTech. So our platform's based on the self-amplifying mRNA, which is, not the conventional mRNA, and explaining along the lines of that, the challenges that come with it. Are there the impurity profiles different? Is the manufacturability different? Along those lines, explaining things is where we are trying to, share information and establish the guidance for the different regulatory agencies.
Yeah, and I would just add real quick, the other piece to consider, though, is the lipids as well. I think you're going to see a little more scrutiny on, do you treat them like excipients? Well, sort of, kind of, but maybe, right? So I think there's a little bit, because a lot of them are custom lipids and things like that. So you wouldn't treat them as excipients, but then they are. So a lot of those kind of things, there's still a lot of work to be done, I think, around the overall schematic of getting future mRNAs to market.
So we touch on a little bit about the process and manufacturing challenge for mRNA. I think one very critical and interesting part that we have not touched upon throughout the workshop is around tech transfer. And I do think that with the panelists here, we have our CDMO representative, and we have our sponsors, and specifically sponsors who have gone through clinical trials in later stage now. So I would ask this panelists to give their thoughts around what are the critical parts of tech transferring an mRNA process, drug substance, drug product, from the sponsor's perspective as well as CDMO's perspective.
Sure, I can start off on this because at Arcturus we have a lot of the R&D capability that we can even manufacture at the scale that we transfer to the CDMOs, but we don't necessarily want to invest in having a building and to be able to have the support systems that make it to thrive. So like I briefly touched upon in the interaction earlier, a lot of the times we want the CDMOs to have an open mind and to be able to bring in the process and the people with a lot more flexibility. It gets very hard when the receiving side is saying hey, this is all we can do, this is what we can do, this is from whom you need to buy. And having that flexibility for us was very important because this was sort of a new field, and some of the components that we were getting was not available earlier. And we had trust in our suppliers, and we wanted them to be onboarded with the CDMOs, and to be able to have that approach of, hey, let's get you inside the door, let's learn through the challenges together, and let's implement this at the pace you need it to be implemented. Having that kind of mindset for a lot of the options out there is good.
Yeah, there's a couple different aspects, I think, that so part of, at least, so the lipid nanoparticle process is fairly secretive in a way. It is proprietary. There are a couple of ways to do it, but it was an area where Moderna especially wanted to protect it. So we were very cautious in who we were working with to manufacture that. So we tried to limit it. So we used Lonza as a single drug substance manufacturer for us. So that limited that kind of potential that we would lose the IP that we wanted to maintain. So that was an aspect of it. The other, I think the challenge is that mRNA is not biotech processing. It isn't. mRNA and LNP it's similar. It's using a TFF, it's using a chromatography, but it's not the same. And you have to start, and the things that are important for a mAb production aren't necessarily, they're not a consideration on a mRNA process or an LNP process. So and then if you start looking at the drug product, it's a temperature sensitive product, and so getting your CMOs to understand, look, I can't give you a 4-day process duration. You have to get this. You've got to do cold as best you can. You've got to, there's a lot of those. That kind of information. The other thing, and I mentioned to these guys when we were prepping for this a little bit, was we sent a lot of people all over the world. And so, as an example, we sent a team of 6 to a CMO and to Lonza in Switzerland. So a team of 6. I worked in contract manufacturing. The most anybody ever sent me was a single person. They didn't send 6. So we sent a team to these sites to to train them to, yes, you could do videos, we didn't have time for that. If you had the video, sure, but we didn't have the video, so the next best thing, you send people all over the world. When Lonza here in Portsmouth, New Hampshire was starting up their process, we had one of the process development scientists who scaled up the process, and he was on the floor. We had our early scale process development in our manufacturing facility in Norwood, as an example, to train our own operators on the sensitivity to understand that you couldn't take your lessons learned from recombinant proteins or monoclonal antibody. There were new lessons, and so that was part of it. I think that ended up being probably the success was really, we had people that were there that could say, and choosing partners that were willing to do that, willing to have that kind of relationship, because not every CDMO wants you in their shop like that. So we had to do that and say, look, that's how we're going to run this. And I think because it was a pandemic and you have the luxury of that, you could kind of force some of those things.
So as a contract development and manufacturing organization, we do tech transfer as a standard SOP. So we have a standard SOP, we do it all the time. And even if we are starting from a sequence, there is always a tech transfer of data and information. If it's at a cell line for pDNA, there's a tech transfer, material transfer. I think the biggest thing for tech transfer is having the process, understanding and assessing where your gaps are in information and materials at the start of a tech transfer is very critical. Part of the project manager's key job is to identify that. But also just having not only the flexibility, so we have a platform process that we can adopt a process to, but we're not going to force a client to do our process. We will tech transfer their process in. In addition to that, if they have high value raw materials that they get from a certain supplier that they have a license to, we'll work with that. We're agnostic. We're not going to force them to use our materials or our materials of choice. We'll let them, pick their own materials. But I think the biggest thing during tech transfer is that assessment of what technical and materials you have to start the tech transfer, then actually assessing what you're transferring and the process, and being what I would say open-minded as a client to a CDMO, that maybe what you think is a robust process may not be a robust process. So if we're getting something in, especially from an academic setting, they think it's robust. We get it in and we're like, oh man, what is this? And so, how do we then take that process and create a batch record that then can go into GMP and can be reproduced time and time again? So that, there is a transition from a research process to a GMP worthy process. And so that tech transfer is critical to identify where those gaps are. And then we can fill them and, be open with the client and say, we might need to tweak this a little bit or we might need to improve it, and this is how we can do it, and then get the paperwork to support it. Make sure that batch record is reproducible, that batch record is going to be proven in a non-GMP batch setting, so that you can find out where you're at scale, where your batch problems are, where your gaps are, and then you can close them so that batch record, when you do GMP, is not going to have out of specs, it's not going to have deviations, it's not going to have, whatever. So it is more reliable as a GMP batch record.
Great. And just
one more comment on that. I did want to mention as well, one of the other challenges that we kind of forced on ourselves, but I think if we hadn't, we would have struggled during the pandemic, which was we were using electronic batch records already in our clinical space. So when it became time to run COVID and then tech transfer those processes to our CMOs, we transferred the MES records with them. So it was a little bit definitely unique type of a tech transfer or a relationship with the CMO that we encountered. And I think Moderna, for the most part, that was kind of how we had worked with CMOs previously. We didn't have much of our own filling, so we always used filling CMOs, and we challenged them to do things the way we wanted them to do things. So that might be MES, that might be, and that I can't imagine going through that much paperwork if we didn't have electronic batch records.
Yeah, and just a follow-up question for Molly. So you've probably seen a divergence of processes when a customer come in. How as a contract development and manufacturing organization, how can you do to make sure you can accommodate all those and make sure the implementation kind of fits into your own work, so that there's not too many redundancies or miscommunications or mixed up. And the other thing is, do you see a convergence of processes, or is it still pretty divergent that people are using different technologies throughout different customers?
Well, I think the BioNTech, Pfizer, and the Moderna platform is duplicated a lot because it is not, the proprietary stuff's still proprietary, but what's published is published. And in the open domain. So I do think that you see that kind of platform often, but there are different technologies that are coming out. There's the circular RNA, there's the 2 RNA in 1 LNP, there's the whole PCR versus plasmid, footprint for your IVT that is kind of a new and maybe still needs to be proven. But as a CDMO, you have to stay innovative in everything you do. And so we actually have innovation projects that we're funding where we have people testing out different processes and technologies and equipment. So we're looking at Quantum's equipment, for example, give them a little plug, and some other ones that are out there for automating processes, and so that we just got to keep on the cutting edge, and it takes some research, it really does, but we are seeing the whole gamut of different types in saRNA, tRNA, anything but the chemical synthesized oligos we're seeing. So you do have to, and some you just say, yeah, we can't do this. We don't have the equipment, and if we wanted to invest in the equipment, this is the CapEx that it would require. Mr. Customer, can you pay for this? If yes, then we can move forward and do the tech transfer. If not, then you might have to find another supplier. So those are kind of a hard conversation to have, but you do have to have them. But staying on the cutting edge is really critical, and using automation as much as possible is going to be in our future. The electronic batch records for sure is something that we'll be adopting as well. But any robotics or automation, predictive AI, I think is probably something that we're going to have to look at for process, just looking at process parameters, and the PPQ, I think eventually is going to be all AI driven at some point, but that's a prediction I make for the future.
Cool, thank you. I also wanted to open up to the audience, so feel free to raise your hand if you have any questions. I think we can have a more detailed discussions. If there's any particular questions that the audience would like to ask, we can go towards that direction.
I'm interested in hearing your perspectives on pain points and opportunities. Sorry about that. I'm interested in hearing your perspectives on the pain points and opportunities in the purification processes for mRNA, particularly the fact what emerging structures, circular ones, and so on, are the things that are kind of challenging for everybody.
So there's traditional chromatography, and then there's HPLC-based separations, and then there is the more recent ones where there is like monolith columns and other things, right? Like there is a lot of these technologies out there, and to be able to like evaluate their usage for separation, I think if a lot of universities, they tend to like publish papers, that knowledge can also like help to reduce that learning curve. For example, right now, earlier this morning, we were talking about how double-strand RNA is like a big thing in mRNA production, right? And if more and more publications come out where they support and show that, hey, this is how you could potentially reduce it, either to begin with, like have less double-strand RNA, or to be able to like, go ahead and show how you're like separating the different species that's produced in the reaction. That's where I think it'll benefit the industry. There's also the IP side of things that companies do want to monetize. That's why the thing is, academic institutions, you're able to like publish things, then that information will reach around faster.
Yeah, I think on the especially on the mRNA side, because again, if you don't have the double-stranded RNA, then you don't have to worry about it. But you're making choices in your IVT reaction. Typically, when you're eliminating the double-stranded RNA, you're probably making some yield hits, or you're making other sacrifices for it. Right? Nothing is free in that space, so it's balancing that. I think as a couple other conversations went on today around the different enzymatic reactions and different ways of doing co-transcription and things like that, that's going to introduce new challenges. For us, I think the oligo chromatography is affinity chromatography is a great operation, right? It's a single step, and you can manage so many different things. But that might work for vaccines, maybe. But when you look at therapeutics, and maybe it doesn't. And then I think if you start thinking about like on the mRNA side, I think most of that's going to be out there pretty soon. Most of the people, you can see the technologies going in that direction. I think the LNP space, me personally, I don't know if it's age or just having gone through the pandemic, but I'm a little altruistic to think that, hey, look, can we accelerate that tribal knowledge instead of trying to keep it to ourselves? But that's not for me. That's my IP lawyers tell me what I can and can't say. But I think that's an area where I think one of the things that I always want to sort of make sure I get across is that what BioNTech and what Moderna has done is showed so many other companies that it's possible, right? Before that, if I said I have to scale up the LNP process, somebody would have said there's no way, you can't do that, and now you can, and we can tell you can. I think about if you go to monoclonal antibody, is there any particular reason why we stopped at a 2-meter column besides the fact that it's 2-meter column? Yes, probably not, but there are some limitations that happen during monoclonal antibody processes where you had a particular resin, you got to 80 centimeter, 1 meter, and that was it. You couldn't scale that up because it was going to fall apart in the column. So I think there's going to be a limit to LNP probably. But somebody's got to find it, and is our scale close enough that we feel confident that we're not going to scale it up again? No, probably not. We want to look at other things, continuous manufacturing. I think that's the next, as much as continuous manufacturing is the next for every other process, that's going to be the next thing for mRNA as well. Because I think we can when we start looking at the number of drug candidates, and if anybody listen to our CEO, he's talking, we're in the hundreds in the next 10 years. That's a lot of drug candidates. We have to be able to go faster, more products faster, less cost, less variability. So I think those continuous manufacturing processes would be, if I had to say where should a new company focus, obviously get a product out there, get some money, but I think continuous manufacturing is really a place where you could start to differentiate yourself because you can drop those COGS way down. Optimizing your enzymatic reactions is going to be an extremely important area to get your COGS down. I think those are areas to focus on. Right? We know that the enzymes are expensive, so you got to figure out how to either make them cheaper or use less of them, optimize them. And that could be, like you said, we've talked about fed batch processes. That's a way to optimize your components as well. So look at those opportunities.
So I think there's, per unit of operation, the pDNA, the IVT, the LNP, there's purification on all 3 steps. And I think they all have different challenges and different scale up or scale out challenges per. So, luckily plasmids, you don't need that many of them because if anybody's ever worked with plasmids, those buggers are sticky. And they're very difficult to purify. But there are some more innovative resins that are out. There's some membrane technology, like nitric separations, that has some mixed mode and some good carboxylic acid chemistries that can really work well with plasmids. So I think that I think innovation is going to be in the resin side for plasmids in particular. When you get to IVT and to the LNPs, then it is more about scaling. and how can you scale it using the existing current technology? How do you get to the cycling and the continuous manufacturing? And again, it's really looking at the TFF, the possible membranes and things like that to do it. So it's a challenge at each one of those unit operations, to include the 0.2 micron sterile filtration, because things get clogged up when they're the size that they are. So it is a challenge, but I think it really is looking at innovation and each unit of operation for each type of purification is going to be the future for it. Did that address your question? Okay.
Also, I just want to add, if you can make lyophilizers much larger and make them, work much more quicker, that'll be your biggest moneymaker.
Any other questions that the audience would like to know?
But I think, we've touched on a little bit, understanding where the double-stranded RNA is coming from. Obviously, if there's an impurity that's causing that, or there's an impurity coming, then that you've got to understand that, right? That's a key thing. For us, I would say the biggest challenge has been the lipid nanoparticle. We had a talk earlier that talked about the adduct formation. And that adduct formation can be problematic. So that's something that, and for us, we rooted that back into the lipid manufacture process itself, which we did the development, but then we outsourced it to a third party for scale up purposes. But we found that adduct formation. It really was something that was happening in their process. So. And we hadn't gotten to the point where we understood that yet, right? We weren't in that place. So very, very extremely quickly, right in the middle of the pandemic, we had to go very fast, and we had to understand where is that coming from. The short version of that story was we just took that one particular lot and put it over there for a little bit while we could go back and figure it out. So, but we had a strong chemistry group, and they were able to figure out that adduct formation was back to the manufacturer, and could figure out a way to a polishing step, if you will, that they were able to put back in. So it's just one example. I think it's always difficult to do your critical raw material aspect. I've been in the industry for a while. People don't start doing your critical raw material analysis till you're kind of late stage in commercial manufacturing, and kind of by there, you're really causing some problems, right? So I think as soon as you can, as the adduct papers well published, and but understanding those a good research about what's out there for mRNA, and making sure that you have an understanding about your, as you said, if you tweak your platform, how to maybe there's a new critical raw material that comes out that you didn't realize was there before. But like I said, for us, and then that was the one good example I had for you.
So from our perspective, it's, if there are organic solvents, for example, in your processes, right? Like, to be able to understand what kind of intermediates are produced. I think more recently across the industry, there has been nitrosamines kind of like evaluation, and a lot of focus on that, right? So getting to understand the details of how your process is made all the way from the beginning, where your suppliers are getting material, and to be able to go down that path to understand, hey, it comes from, for example, Germany or from China or from Netherlands or from any other country like Japan, to be able to go down and trace every single source to attain that origin is very key. And to be able to understand all those intermediate steps to be able to get an idea of what is happening, where it's happening, how it could interact, A can interact with B, can interact with C, to be able to take it down 2 or 3 layers is key. And like Aaron is mentioning, a lot of this doesn't happen until, you say hey, this product X or product Y is going to get you so much revenue, and you want to be able to set aside that much money to do this analysis. And a lot of these analyses are not, cheap. So it's key that as folks that work in this industry to keep an eye out for this and to be able to understand and make those educated calls to be able to whether utilize it or not utilize it, and to be able to communicate that to your vendors and suppliers. For example, to not use any animal origin materials anymore. It's pretty clear from the regulatory agencies that nobody wants to deal with it. And it comes from a good reason. So having an understanding of that from an early stage, from the research development stage, will set you up for success.
Yeah, and I think it's really the impurity profile that I think you're really trying to address here for a safety standpoint. Potency is one thing, and making sure your cell-based assay is good enough and robust enough. But it really is that impurity profile. And one of the things, because we are agnostic to the technology of the materials, we have done a study to look at all the high-value raw materials that are available globally and how do they impact our platform. And so looking at all the different types of enzymes that are available, and there are, some are, the cost variance is all over the place, honestly, and they come from all sources, as he described, all over the world. But how does each one impact your impurity profile of your end product, and really looking at that and seeing what they impact, how they impact, why, and how can you get rid of it if you need to from a process standpoint. So looking at, whether it's a lipid or whether it's an enzyme or whether it's the solvents. I think people were kind of, because there were biologics producers, they weren't looking at solvents as a source of an impurity, but yeah, lo and behold, there's different grades of solvents. So it really ends up, what is that impurity profile? So you really have to take a look at it. And if you're going to substitute something in that is less expensive or more available or has a better supply chain, you do have to look at all of these things to include, as you said, the animal source, and go back and make sure your vendor assurance program is really looking at that base source. It takes time, and we worked on it for a long time. We inherited it from the company that we carved out of. They started working on it about 8 months ago, and now we have a really good profile of materials that we can say, yeah, not that one. These 5, yes. This one, no. So it really you just have to really look at that and backward integrate to your material source.
Yeah, and I would add, because we, like I said, we experienced that in a little bit because we went so fast, we didn't have a chance to go back and look at, say, some of our raw material specs. And when we were getting ready to file our BLA, we're like, this is not appropriate material. So be careful when you're establishing those raw materials for your company to say, it might be okay to have the same material that you want to use in development and in the GMP space. That's okay. But make sure that it's the higher grade, right? And I think where we ran into trouble, we were going so fast. Somebody introduced something as simple as sodium hydroxide. Something as simple as sodium hydroxide was not of the appropriate grade. So now we're ready to get into, commercial manufacturing. We're like, we've got to change this. And so making that change, something as simple as sodium hydroxide. So I think don't build your company to be a phase 1 clinical company. Build yourself to be a commercial company. Yeah, you don't exercise the work maybe yet, but at least understand this is to make the right selections, and then you do the right things at the right phases. But if you start with the wrong selection, you don't find it till you get to the end, and then you're struggling to have to deal with it. So things like that are something as simple as raw material selection. Just you want good control over that at the very beginning of your phase 1 clinical trials.
So I think we have time for one more questions, and then I think we're close in time, but I'd like to open up for one more questions all right, cool. And we'll maybe have 2 panelists to answer them.
To follow up on Aaron's comment about the choosing raw materials for different grade, what is your opinion for when to finalize the materials and the packaging size, when the latest stage to decide what is the materials that is finalized, the phrase, and also the packaging formats? Thank you.
I mean, I would, if you're bringing the simple answer for the grades, if you're bringing material into phase 1 clinical trials, it should be GMP grade material or better. If it's not, which in certain cases that may not be, you might have to use research grade. That's okay. I mean, it's allowed, but you have to understand it. So I would make sure you understand that, because if you do bring in research grade material, you're going to have to do extra work when you get later down the road. So if you can choose between GMP grade and research grade, pick the GMP grade every day. Package size, I don't have much of it. I don't think it really matters. Pick the obviously from a cost perspective, I think it's like anything, right? Buying a 2-liter bottle of soda is better than buying a 12-ounce can, but if it's going to expire, then buy the 12-ounce can, right? So you have to be, I don't think the package size really matters too much, unless I missed that.
Yeah, I can speak about it. So, yeah, I think as he spoke about having research-only product versus a GMP or a food grade. If it's a chemical, go for USP or multi-compendial or a food grade at a minimum. Don't use research grade. If it's a plasmid, go GMP. I spent a lot of time as the chief operating officer of a viral vector CDMO. And we had 3 clients put on clinical hold because they were using research-grade plasmids, and the FDA wanted traceability of those plasmids. They wanted to show documentation of those plasmids, et cetera. And they didn't have it because they were using research-grade plasmids. So if I can recommend anything, and my team knows this, I'm always on my soapbox about this, use GMP-grade plasmids, because you do need that traceability. You do need to be able to show that you have produced them using good manufacturing procedures.
Cool. And Mukunda, since you didn't answer those questions, I'm going to ask you the last questions and we'll close this panel.
Sure.
What was I going to ask? So one thing that I think Aaron actually brought it up during your presentation is about a platform approach, right? Having a platform approach potentially could accelerate some development activities. I like your thoughts around, some of the things that you still cannot skip, even going with the platform approach, something that, might be overlooked by some others, skipping those activities, but ended up being, a trouble. So, sorry, this is a hard question, but I like your thoughts.
So we are still going through this process of a platform approach, right? Like, it's very key to explain in your validation master plans and the other documents that you use, be it like the manufacturing process control strategy, or for that matter, the other cleaning validation documents, or any of those kind of situations. You have to explain what is it that you're looking for with that particular process qualification. And if you say that, hey, you want to bracket the scale, or you want to bracket molecules, or if you want to bracket certain purification strategies, it's necessary to describe ahead of time and let it be a very transparent approach for you to make it a platform. For us particularly, you don't want to go through this qualification processes multiple times, and that's what I think Aaron was also mentioning during his talk. It's key to describe that. And also one thing that I've seen in my experience is if you put in a purification step, it's hard to remove it. So before you put in, you may want to do a little more characterization and spend time 3 months, a quarter, or 2 quarters, if you think that that purification step is not needed in the future, I would rather invest time ahead of time rather than, go ahead and, spend countless hours trying to justify why you want to remove it in the future.
Yeah, I would just, I know we're out of time, but just real quick on that, like I mentioned, I was through almost all of our health authority inspections, and I would say the biggest thing that we missed and we didn't document very well is why. Why did you do it that way? Why did you do that? We knew why we did it because we did it. We made the choices, but we didn't have that justification. So I would say when you make those decisions, have the justification and think about if an auditor asked me, should I have some data? Do I have some data to support that justification? And at the end of the day, my belief has always been in the industry is if you have data that supports your justification or your rationale or your risk assessment or whatever it is, if you have data and you can, you're going to be okay. Is every inspector going to like it? No. You're going to get, but if you have that data, it's hard for them to fight you on it, to be honest, once you have that scientific data.
Well, it's really a pleasure to have you all and to discuss on these topics. I think we discussed a lot, and obviously we don't have a lot of time. I'm sure if we go on and on, it'll never end. But thank you all, and please join me to thank our panelists today for this panel discussion.